Alright, this is a continuation of the meiosis notes. So we just got done talking about how meiosis is important to make gametes. It makes the sex cells, and the sperm and the egg. And the important part is to create variation and chromosomes with less than 23 or less than 46 chromosomes. So let's just quickly review that real quick. So again, you start with 46 chromosomes. They're all paired, 23 pairs. We double them, and then we split twice to get down to 23 chromosomes. These are called the gametes here. I forgot to add that in the last section. And so if this is a female, these are all going to be eggs. If it's a male, we just change these, and these will be sperm. All right, and then these guys in one of these will be the lucky guy to be used for fertilization to make the baby. All right, and same with the egg. One of those eggs would be used. So one from the male, and then there would be another egg over here with 23 to be used for fertilization that we'll talk more about in the second section. All right, so now if we look at variation, so the first part is independent assortment, and each chromosome pair, for each one chromosome goes to each cell. All right, so one chromosome, each pair, one goes to each cell every time meiosis occurs. So what that means is, now I'm going to use some different colors here. So we have, I'm just going to do 4 chromosomes. So let's say I have a pair of, well, let's do 6. We have one, well, we're going to do 3. Because that's all, well, actually, we'll do 4 because that's all the colors I have. So one of each color there. Alright, so our cell first multiplies the chromosomes. 1, 2, 1, 2, 1, 2, 1, 2. Alright, now let's just, well, actually, this isn't exactly going to be the best way to show this. So I'm going to have to change up, so ignore this. Alright, so we have, let's just talk about 2 chromosomes. And on one chromosome we have this big letter A. Well, you'll learn more about that later. It's a gene, and this is another type of a gene. Alright, so we have a big A and a little a. And then down here we have another set of genes. Down here we have a big B and a little b. Alright, and then we have another set of chromosomes over here. And let's say this one has a big H and a little T on one of them. And then on the other one, this has a little H and a big T. Alright, so we go through and we copy our chromosomes, and then we divide once, and then we divide again. So now I'm just going to show you the final products because that's the most important. Well, actually, I'll show you. We'll do the doubles first. So there's going to be one with a big H and a little t. There's going to be another one with a big H and a little t. There's going to be another one, little h, big T, another one, little h, big T. And then over here, our black ones will be, there'll be 2 with a big A, big B, big A, big B, and then 2 with the little a, little b, little a, little b. So now what independent assortment means is these guys can go in we can get various different combinations. So we could get one cell, remember we'll get one from this pair and one from this pair. We're going to get one cell that might have big A, big B on the black chromosome, and then this one might get one of these chromosomes, the big H, little t. Alright, so that's one possibility. The other possibility is, okay, so we used one of each of those. Now this one could come over here again, so this cell could also have big A, big B. But now instead of getting another one of those, we could get down here. So now this is a totally different cell with a different combination of genes. All right. So they have, these guys have these two genes the same, but these two genes would be different. All right, so this guy has a possibility of passing down these sets of genes or these sets of genes. And then we got two more. Now we use both, one of those and both of those. Now these two cells have to have the little a, little b in it. All right, but the other chromosome would be, one would be the big H, little t, and the other one would be the little h, big T. All right, so what happened is we get 4 different cells, and they're all different just because the chromosomes rearranged. Instead of getting all our cells have big A, big B, big H, little t, and that's the only thing he could donate, he could donate a whole wide assortment. And this is only with 4 chromosomes. Imagine if, for real life, you have 46. All right, so that's why brothers and sisters can actually look so much different from each other, because of those wide different assortments of genes you can get. And we're only talking about 1, 2, 3, 4 genes. You're talking about thousands of genes. So that's why brothers and sisters can look so different. All right, so that's the one example. The second one is crossing over. All right, so crossing over is, if we just had that, we're just going to use the 2 chromosomes again, and what one had a big A, big B, and the other one had a, little b. All right, so they duplicate. Big A, big B, big A, big B, little a, little b, little a, little b. Now, before they divide, what happens is they can cross over. So, to show you what crossing over means, they could have, these 2 cells could switch these 2 genes could switch places. The chromosomes actually break off and switch places. So now, instead of having big A, big B, we would have little a, big B over here, and over here this would be big A, little b. All right, and then this one we could maybe just keep the same. Maybe this one doesn't cross over. Oh, sorry, that should be a little a. All right, so they didn't. This one didn't cross over. So now, instead of, so in this scenario, I'm just going to draw the final 4 cells. In this scenario, we have big A, big B, or little a, little b, or big A, big B, or little a, little b. So since there's only 2 chromosomes, I only have 1 in each. All right, 1 chromosome in each. And there's only 2 possibilities. There's either the big A, big B, or the little a, little b. Now, when we crossed over and we make our 4 cells, now we have the possibility of having little a, big B, big A, little b, big A, big B, and the last one would be little a, little b. So again, we've still made, this was started with 2, so we still only had 1 chromosome in each. But now this time, this is different, this one's different, this one's different, this one's different. There are 4 different possibilities instead of 2. All right, and again, this is only with 2 chromosomes and 2 genes. Now imagine if you had, again, 46 and thousands of genes. Then you would have millions of possibilities. Again, that's why brothers and sisters who have the same parents can look so different, and that's why there's so many differences. So crossing over is just chromosomes will, so crossing over. All right, the reason why they call it crossing over is because these 2 actually crossed and changed genes. So they exchange genes or parts of the chromosome, either one, by swapping parts of the chromosome. All right, so that is it for meiosis. So again, for meiosis, you need to understand that it creates 4 different cells called gametes that are used in sexual reproduction and have half the number of chromosomes. And it creates variation, and one of the key ways it does that is through crossing over.